Welcome to the deep dive. Today, we're plunging into the really intricate world of gene editing. As you know, it's a field that demands this massive understanding of complex biology plus cutting-edge tech like CRISPR. The sheer amount of knowledge needed just to design and, run effective experiments, it's honestly staggering. Just think about the hours, the weeks scientists pour into sifting through data, planning meticulously, troubleshooting. It's incredibly high stakes work, very knowledge intensive. Okay, so let's unpack this a bit. Imagine having like a brilliant co-pilot right there with you in the lab, a guide that not only gets the unbelievably complex world of gene editing, but can also help you navigate it with unprecedented speed and accuracy. That's exactly what we're diving into today with this fascinating new system called CRISPR-GPT.
Yeah, it's truly an exciting development because, while these large language models, LLMs, they've shown amazing capabilities everywhere else, they often fall a bit short when you need that really deep domain-specific knowledge for, say, accurate biological problem solving. And it's not really a fault of the LLMs themselves. It just reflects the unique challenges in biology. You've got the inherent variability of living systems. Biological data is often noisy, messy, and the skills and tools needed, highly specialized. So CRISPR-GPT is specifically designed to bridge exactly that gap right in the biological domain.
Right. So our mission for this deep dive then is to really explore how CRISPR-GPT steps in as this AI co-pilot, how it automates and enhances gene editing design, data analysis. We'll look at the real-world impact it's already having in labs, and this is crucial, the ethical questions it brings up, because it's such powerful tech becoming more accessible. Now, we've touched on the complexity. Can you maybe elaborate a little? Why is gene editing such a knowledge-heavy challenge that needs this new kind of AI? Like, what's been the main bottleneck maybe for researchers that CRISPR-GPT really hits head on?
Absolutely. So the core problem really is that doing these experiments well demands a deep kind of dual understanding. You need to understand the CRISPR tech itself and the very specific biological system you're targeting. You can't just, throw a general AI at it and hope for the best. It's just too nuanced. General purpose LLMs, yeah, they know a lot, but they lack that granular, really in-depth biological understanding. Living systems vary like crazy. The data we get is often complex, sometimes messy. Plus, the skills, the tools in biology, they're very specific. It's not like general coding or writing, and that's precisely where CRISPR-GPT comes in as a targeted solution. It's an LLM agent system, purpose-built to automate and enhance CRISPR gene editing design and analysis. We often call it an AI co-pilot in genome engineering.
AI co-pilot. I like that. It fits perfectly. And the range of things it can do sounds, well, massive. It seems like it helps with almost everything. Selecting CRISPR systems, planning experiments, designing those crucial guide RNAs, the little molecular compasses directing the machinery, right? And choosing delivery methods, drafting protocols, designing assays, analyzing the data.
Wow. So it sounds like it could potentially be an end-to-end assistant for a researcher, handling pretty much every stage.
That's the goal, yes, to be that comprehensive.
Okay, that sounds incredibly powerful, but let's look under the hood a bit. How does this AI co-pilot actually work? What gives it that specialized smarts? All right, this is where it gets really interesting. CRISPR-GPT uses the core reasoning abilities of LLMs, but in a very structured, almost collaborative way. It's good at complex task decomposition, breaking things down, making smart decisions, and enabling this interactive human-AI collaboration. The key insight isn't just its AI, it's that CRISPR-GPT acts more like a highly specialized scientific team. Different AI agents handle different roles. Think of it like, okay, you have an LLM planner agent. That's your project manager coordinating everything. Then a user proxy agent talks directly to the human user. Then you have tasked executor agents doing specific steps, and tool provider agents connecting to external software, huge biological databases. And all these parts interact smoothly, passing information decisions back and forth, like a well-oiled research team. And crucially, it doesn't just rely on general knowledge floating around the internet. It pulls in expert knowledge from top practitioners, from peer-reviewed papers in gene editing. That's done using a technique called retrieval augmented generation. So its advice is grounded in real, current science. It also uses chain-of-thought reasoning, which means it can break down really complex requests into a sequence of distinct, manageable tasks. And importantly, it keeps track of how those tasks depend on each other. Now, you asked how it learns to think like a scientist beyond just facts. Well, here's a really cool bit. A key part of CRISPR-GPT is a specialized LLM that was fine-tuned on real scientific discussions, specifically about 4,000 discussion threads collected over 11 years from a public Google group just for CRISPR gene editing. That unique data helps it genuinely think like a scientist when tackling biological problems. It gets to the nuances, the common hang-ups researchers actually talk about.
Wow, that's incredible learning from actual scientific back and forth from real researchers hashing things out. It sounds like it can really adjust based on who's using it. You mentioned tunable levels of automation. How does that work for, say, a beginner versus a seasoned pro?
Yeah, it offers three distinct modes. First is meta mode. This is really designed for researchers just starting out. It guides them step by step through all the essential tasks from picking a CRISPR system right through to analyzing the data. It's very interactive, gives clear instructions, asks for clarification at each decision point, almost like having a mentor walking you through it. Then there's auto mode. This is more for advanced researchers. Here you can just type in freestyle complex requests. The LLM planner automatically breaks it down, builds a custom workflow, and runs it. It's smart enough to fill in missing details, and importantly, it explains its reasoning. So the user can monitor, adjust if needed, they stay in control. And finally, there's the Q&A mode. This is for on-demand scientific questions about gene editing. Here, CRISPR-GPT pulls together information from multiple places. It uses that unique, fine-tuned CRISPR Llama 3 model, the one trained on the discussion data. Plus, it uses RAG to pull from about 50 key curated papers and also general LLMs to give really comprehensive but concise answers.
Okay, so whether you're brand new and need that hand holding, or you're an expert just wanting to speed things up, CRISPR GPT adapts. It really helps you get the specific knowledge and guidance you need at that moment. The flexibility sounds like it could genuinely change how gene editing happens in labs. But, seeing is believing. What actually happened when they took it out of the simulation and into a real wet lab? Did it work?
Ah, yes, this is where the rubber really meets the road. And the evidence is pretty compelling. They had two junior researchers, both unfamiliar with gene editing techniques, use CRISPR-GPT as their AI co-pilot for actual experiments. So demonstration one, they aimed for a multi-gene knockout. Pretty ambitious. The researchers designed and carried out a knockout of 4 genes, TGFR1, SNAI1, BAX, and BCL2L1. This was in a human lung cancer cell line, A549, using CRISPR-Cas2la. Cas2la is often good for targeting multiple genes cleanly. The results were remarkably consistent. Around 80% high editing efficiency across all 4 genes, confirmed by next-gen sequencing, the gold standard. But it wasn't just about the edit efficiency, which is great, but did it do anything biologically relevant? So they did biological validation. For the TGF1 and SNAI1 knockouts, these genes are key players in cancer spread. Functional tests showed up to a 9-fold drop in CDH1 expression and a huge 34-fold drop in VIM expression. These are really critical markers in what's called the epithelial mesenchymal transition, or EMT. It's basically how cancer cells learn to metastasize, become invasive, so a big impact there.
Okay, second demonstration. Same junior researchers guided by CRISPR-GPT. This time, they epigenetically activated 2 genes, NCR3LG1 and STIG-IM1. These are involved in resistance to cancer immunotherapy. They did this in a human melanoma cell line, A375, using CRISPR-Cas9, which is used for activation, not cutting. Again, success. Up to 56.5% activation with NCR3LG1 and a really high 90.2% for STIG-IM1.
That is astonishing. Both experiments worked first time with researchers new to the technique. Anyone who spent, long nights in the lab trying to get gene editing to work would find that success rate almost unbelievable. What was the reaction from the team, seeing these junior researchers get such results so fast?
Oh, absolutely. It was a powerful validation showing how systems like this could, democratize access to these complex methods. Now, what's really interesting is why CRISPR-GPT does so well where general LLMs might struggle. It's not just having broad knowledge, it's this specialized intelligence that truly gets the nuances of gene editing. They tested it rigorously using a benchmark called the gene editing bench. And CRISPR-GPT clearly outperformed across key tasks. Take experiment planning. It scored over 0.99, basically perfect on accuracy, precision, recall, F1 score. Significantly better than general models like GPT-4o at laying out complex editing tasks and managing how they depend on each other. Then, delivery method selection. It had a big advantage over baseline GPT-4, GPT-3 turbo models, especially for the tough cases like hard-to-transfect cells or primary cells. These are areas where general LLMs often trip up because they lack that specific biocontext. For gRNA designing those guide molecules, it accurately picks the right design actions, configures the settings. And here's a really specific example that shows its deep biological reasoning. In one cancer study, CRISPR-GPT specifically chose key exons, exon 3 and 4, for targeting the BRD4 gene. Now, other common tools, like CRISPR or ChopShop, they would have suggested guides hitting non-essential exons. Those guides probably wouldn't have worked, wouldn't have produced a biological effect. So CRISPR-GPT isn't just following simple rules, it's reasoning biologically about which parts of the gene actually matter. And even in its Q&A mode, it did significantly better than GPT-4 at 12% more accurate, 15% better reasoning, and impressively, 32% more concise for advanced questions. General LLMs sometimes made factual errors or just gave these long, rambling answers that weren't totally relevant. Like, There was a question about fixing cell growth problems in MCF7 cells after Cas9 editing. GPT-4 gave a long list, but a couple of points didn't even apply to MCF7 cells. CRISPR-GPT gave a concise, accurate summary tailored specifically for that cell line.
Right. So the big takeaway here is CRISPR-GPT isn't just a jack-of-all-trades AI, it's a specialist. It genuinely understands the nitty-gritty of gene editing and can apply that deep knowledge effectively. It's not just about having tons of info, it's about having the right info and knowing exactly how to use it in a biological context. Makes sense.
Exactly. But despite all these strengths, we need to be realistic. CRISPR-GPT, like any advanced AI, it does have limits right now. Its performance really hinges on getting high-quality instructions and that discussion data from human scientists. But getting enough of that top-tier data, well, it's really hard to collect and scale up effectively. Also, just evaluating tools like this is inherently tough. You need substantial, detailed feedback from expert biologists to truly gauge its capabilities and where it needs work. Currently, its gRNA design is mostly focused on human and mouse targets. That limits how widely it can be applied across other species for now. And yeah, like any AI, it sometimes struggles with extremely complex requests or very unusual biological situations that are outside its main training.
That's a really important point. Even with these huge leaps, AI development is still a journey, right? There's always room for improvement. The goal isn't instant perfection, but strong, iterative progress. Okay, now shifting gears a bit, but this is critical. When we talk about tech this powerful, we absolutely have to talk about ethics and safety. CRISPR itself, and now AI tools like CRISPR-GPT that make it easier to use, they bring up some significant considerations we need to tread carefully. How does the system handle these complex issues?
Yeah, this is a vital question. Getting into responsible innovation, there's definitely a dual-use potential, meaning the technology could, theoretically, be misused for harmful things, like creating bioweapons or engineering dangerous viruses. And then there's the huge ethical issue around editing human genomes, especially germline cells or embryos changes that can be passed down generations. That raises profound ethical concerns, and it's illegal in many places, including the U.S. So it's crucial to know that CRISPR-GPT has robust safety mechanisms built right in specifically to mitigate these risks. For instance, users absolutely cannot bypass specifying the organism they're editing. That's step one. If the target is human tissue or organs, the system immediately flags it with a clear warning. It gives a direct link to the International Moratorium on Heritable Genome Editing, explains it, and this is key, requires the user to confirm they understand the risks and guidelines before they can even proceed. And even more strictly, if a user tries to request something involving editing human germline cells or working with dangerous pathogens, CRISPR-GPT just triggers an error message and stops. Full stop. It refuses to assist with those requests. Data privacy is another big one, especially with potentially sensitive human genome data. To prevent sharing identifiable private human or patient sequences, the system is designed never to store long genome sequences on its server. It also has a filter that scans prompts for any long DNA sequence, 20 bases or more, before sending anything to external LLMs. If it detects one, it flags an error and tells the user to remove the sequence manually.
Okay, that is incredibly reassuring to hear about those specific safeguards being built in from the start. Critical thinking is so vital today with information overload, and especially when we're dealing with tools this powerful. It really shows thoughtful design, having those mechanisms in place to guide its use responsibly. So looking ahead then, what does all this mean for the future? Where does CRISPR-GPT or technology like it take scientific discovery next?
Oh, the future potential here is really exciting. We can definitely expect further expansion. Connecting CRISPR-GPT to the latest advances in, say, genome and protein foundation models, sophisticated plasmid design tools, other machine learning models. This would let it help with experimental design and analysis way beyond just gene editing, broadening its scope across biology research. But maybe the most mind-blowing prospect, the thing that feels almost like sci-fi, is the potential for integrating CRISPR-GPT with automated lab platforms, robotics. This could enable genuine end-to-end automated experiments, minimizing manual work, dramatically speeding up the pace of discovery. Quite something to imagine.
Wow. Yeah, automated labs run with AI guidance. That really is mind-boggling. It truly feels like this is just the beginning of AI's role in transforming science, opening doors we can barely even picture right now. And that wraps up our deep dive into CRISPR-GPT, an incredible AI co-pilot that's already proving its value in the complex, high-stakes world of gene editing, making these intricate experiments more accessible, faster, and importantly, with built-in ethical considerations.
And maybe a final thought for you to consider. As AI tools like CRISPR-GPT keep evolving, they absolutely promise to accelerate science beyond what we can currently imagine. But at the same time, they put this new, higher premium on our human values, on ethical oversight, on collaborative vigilance. So the big question for all of us is, how will we as a society make sure this incredible power is used responsibly, truly for the benefit of everyone?